Greetings, everyone. This is Ken Mandel, Director of the Computational Health Informatics Program. Many of you have joined us previously at our Landmark Ideas Series and other presentations that we've made available more widely in the community as the world moved to Zoom for webinars, and we've been very pleased to have a community forming around some ideas. Today, we are launching a new series within our offerings that we call Frontline Dispatch. And today, the Frontline Dispatch series deals with issues of more urgency so that we can be responsive to changing conditions. And as folks know, that has been a very frequent occurrence, rapidly changing conditions where new information is necessary. So the Computational Health Informatics Program is a 27-year-old program here at Boston Children's Hospital. We're affiliated with Harvard Medical School, and we work across population health, clinical care, research and discovery. And today, I'm very pleased to have one of our own faculty members, Ben Reis, a faculty member in the Computational Health Informatics Program, and Professor Ben Reis is also a faculty member at Harvard Medical School in the Department of Pediatrics. Today's talk, and you may have heard Dr. Reis speak previously about the Israeli study of the Pfizer vaccine that was published in the New England Journal in one of the most high impact papers in recent memory. Today, he's going to deliver an address to us called Dispatch from Israel, COVID-19 Vaccines and the Delta Variant, What We Know So Far. And I will turn it over to Professor Ben Reis.
Thanks very much, Ken. So we're just going to share a screen here, see if we can get this going. All righty. Okay, so can you see my screen? Ken, can you give me a confirmation that you can see the presentation?
Yes, we can see you.
Okay, great. So thanks everyone for joining. I want to thank Ken for coming up with this idea of the Frontline Dispatch series and asking me to speak today. What we're going to do today, since we have a large and diverse audience with different backgrounds from around the world, is we're going to spend the first maybe third of the talk understanding what the Delta variant is and what we know about it so far. The second third of the hour, we're going to look at the story in Israel and what's been going on in Israel, both before the Delta variant and now during this current wave, which is mostly due to the Delta variant. And then we're going to try to save the last 15 to 20 minutes to answer your questions. And those questions you can put in the chat, and Ken is going to moderate. So let's get started. Okay, this is planet Earth, and as people who saw the previous talk may remember, this globe has been turned upside down by this spherical object, the SARS-CoV-2 virus. But today we're going to, instead of talking about the vaccine and how effective it is against this virus, we're going to look inside the virus, and we're going to see what lies behind these little red spikes. So by now, probably everyone has heard about these spike proteins that cover the virus and their role in infection. But we're going to take a deep dive. And the reason we're going to take a deep dive into these spike proteins is that's going to be necessary for us to understand what is a variant, what is a mutation, what is different from the, between the Delta variant and the Alpha variant, et cetera. So if you were to crack open a SARS-CoV-2 virus, a coronavirus, you'd see that on the outside, you have these spiky proteins sticking out. They look like perhaps little trees, more than spikes, or little mushrooms. But on the inside, you have this squiggly thing, which is an artist's rendition of the genetic code, the blueprint for the virus. This is a piece of RNA, which is different than what we have. In our cells, we have DNA, but this is very similar. The genetic code for the virus is in this one is an RNA virus. And in very simple terms, this is the instructions. These are the instructions for building another virus. So the virus is basically a self-replicating machine And its goal is to get this squiggly yellow thing into a human cell or into animal host or whatever host it's infecting. And it's going to try to get that host to build more copies of it so that they can go on and infect other host cells and build more copies of it. So it's a very efficient killer, a very efficient machine. It has the payload, which is a squiggly genetic code inside. And on the outside, it has a protective shell. This is the red circle here. And it has a bunch of spike proteins shown in pink here. And those spike proteins are essentially keys that allow it to get in to the cells that it wants to infect. Okay, so that's the basics of the virus. Here's the life cycle of the virus, which you've probably seen many times in the news by now. The virus here is in red. The host cell, in this case a human cell, is in green. The virus unlocks the door to the cell using one of these little keys, these spike proteins. It gets into the lock, which is in this case is represented by an ACE2 receptor, a protein on the surface of the human cell. Gets into the cell, opens up, kind of fuses with the cell, dumps out its little squiggly RNA, and that RNA takes over the cell's manufacturing capabilities. And those sets of instructions are basically turned into additional copies of the virus that are then sent out of the cell, sometimes to the tune of millions of more viruses. And that's how a virus can very quickly take over a human body. So that is the background for the virus. It's a very efficient self-replicating machine. Okay, what are we looking at here? Well, these are the building blocks. These are called amino acids, and they are the building blocks of proteins. I apologize to people on the call who know all this stuff, but I just want to make sure we have these ABCs down before we go. There are about 20 or so amino acids that are used by the human body to build anything that it wants to build, from your fingernails to your muscles, or to viruses. And these are the building blocks. They're called amino acids. And so the instructions, the RNA of the virus, are translated into these amino acids. And when you string a bunch of amino acids together, they start curling up into these 3D structures, which you see here, creating those 3D shapes that can have biological functions. So back to our picture, this is the virus. Inside we have the squiggly genetic material, and the outside we have the spike proteins. And this is the actual coronavirus. This is the genetic material. It's 30,000 base pairs of RNA. That's it. So those 30,000 base pairs, which are made up of an alphabet of 4 possible letters, they basically comprise the sum total of the information that constitutes the virus. And that's what's inside the virus, 30,000 base pairs. Now those 30,000 base pairs are translated into proteins, as we said, by the human body to build another virus. The part that we're going to look at is this little region called here, labeled S. And that little region comprised of about 4,000 base pairs, so about 4 out of the 30,000 base pairs, which are the letters that make up the genetic code of virus, actually code for the spike protein, so those little pink things that we saw sticking out of the virus. And when you translate those into proteins, you end up with about almost 1,300 proteins, a sequence, if you will, a long string of 1,273 amino acids, one after the other. And when you translate the genetic material into that protein sequence and you let it curl up, it curls up into a spike, curls up into that little mushroom shape that we saw. And what's really interesting is that the entire world scientific community has been focused on this virus for the past almost year and a half or more. And prior to that, we've been studying coronavirus. So we actually know a lot about the structure of this protein and what each individual base pair does, or if you will, each sequence of amino acids that are translated from the base pairs, what they do. And scientists have been able to focus in on this little region right here of a few dozen amino acids that are known as the receptor binding motif. This is the business end of the spike protein. This is the edge of the key that fits into the lock. And the numbers that you see here are which number amino acid, which position along this string that creates the spike protein. So we actually know pretty well what is the business end of this spike protein. And what happens is once the genetic RNA of the virus goes into the cell and creates this string of proteins, it actually folds up through the laws of atomic physics into this little mushroom shape that you see here. And you see that this little RBD, this receptor binding domain, ends up at the tips of the mushroom, the tips of the spike. And this little red area is basically the tip of the tip. It's the business end of the mushroom. And this is what it actually looks like if you were to zoom in biologically. It's a bunch of curly strings of molecules. Here's another picture of it. And what happens is, this is a recent visualization that I found very illuminating. So at the bottom of the screen, we have the virus with a little spike sticking out. And at the top of the screen, we have a human cell with a little ACE2 receptor, which is basically a lock. Okay, so here's the key, here's the lock. And what happens is the business end of this mushroom, this business end of this spike, fits right in at the atomic scale to this lock. So the key is fitting into the lock. A whole bunch of things happen, and then there's a fusion event where basically the key is inserted into the lock and the cell and the virus are bound as one. And a fusion event happens, and the virus then enters the cell, releasing its genetic material. So that's what's happening. That's how the virus is getting in. And we have a pretty good understanding down to almost to the atom of what these things look like. Okay, so that's the basics. Why did we just go through that entire process? Because we need to do that in order to understand what mutations are and what variants are. So a virus is actually pretty simple. It has this squiggly genetic material. It gets translated into this string of proteins. Those proteins fold up into this little spike, which becomes a key, fits into the lock of the cell, and gets in. Well, what happens if the virus is a bit sloppy in copying itself? Once it gets in the cell starts producing many, many copies of the virus. What if those copies are not exact? And specifically, what happens if one of the genetic letters up here is miscopied? Okay, so the RNA is, if there's 4 letters, we'll call them A, B, C, D for now. If an A is miscopied as a B, or vice versa. Well, that causes a different protein sequence to be made, which causes a different shape to be built. And scientists have been able to actually identify specific one-letter mistakes that have improved the virus's ability to open up this lock. Okay, and so they name these mutations, and there's a whole world of scientists that discuss these mutations. If you look over here, it says N501Y. What that means is that the amino acid N, which is in position 501, has turned into the amino acid Y. Okay, so position 501 is right here near the business end of the protein. Over here, amino acid E, which is located at position 484, has turned into amino acid K, and so on and so forth. And since all of these amino acid changes are located right at this business end, right at this red area right here, they can have massive implications for the virus's ability to penetrate the cell. And what's really funny is these people who spend very long hours staring at these molecules have become friends with these mutations, and they've started giving them nicknames. So the N becoming the Y has become Nelly, E becoming K has become Eek. This one is the most popular name. The D that becomes a G has been nicknamed Doug, and viruses that do not have this replacement are called Douglas because they do not have Doug. So there's a whole world of snarky scientist jokes of people studying this stuff. Here's Kevin, where K becomes N at position 417. But again, you notice all these numbers, all these mutations that we're looking at are right here, right near the business end of the viral spike. Okay, now you'll see that I've started labeling alpha, beta, epsilon. What is that? Well, these mutations, these changes, these single letter changes that scientists have found in these viral strains have shown up in different parts of the world. Alpha is what we now call, what used to be called the British variant. Alpha is a strain that was first became dominant in England. We're not sure if it started there in the Kent area. And it has these 3 mutations, which made it much more infectious. Beta, which used to be called the South African variant, which is something that became dominant in South Africa months ago, had this Kevin mutation. Epsilon, which used to be called the California variant, had the Larry mutation, where an L became an R, et cetera, et cetera, et cetera. And you can see here that each of these mutations is a beneficial one for the virus and a detrimental one for us who are trying to fight the virus. So recently in India, actually not so recently, in September, October of 2020, a new strain of virus was found that contained both the Larry mutation, this thing that we had previously seen in the California variant, and the Teak mutation, where a T became a K, again, in these very important positions that are the business end of the key. There's another version of this, this was found in India, another version of this virus also had the Kevin mutation, which if you remember, Kevin is our friend from here, from South Africa. So this is kind of a worst of all worlds combination, where the all-star mutations that have helped the British variant become so bad, and the South African variant and the California variant, all those horrible mutations that made those viruses so dangerous have come together into a what might be called a double mutant or triple mutant. It turns out the Delta has about 20 mutations that are known to be beneficial for the virus. I'm just listing a few of them here. So this is the world of mutations. Scientists choose to call certain strains, certain families of viruses that spread from one person to another in certain communities. Once there's a preponderance of these viral families that inherit these mutations one from another, they call them different strains. And recently, the WHO started, since there were so many different ways of naming them, I don't know if you've seen these, some of them used to call them using the Pango lineages, which is B117, sometimes you see that in the news. There's a number of other ways to categorize these. So to make life easier for the world's conversations, the WHO and the public health community recently started just calling them by Greek letters. And the first four, alpha, beta, gamma, and delta, used to be called the British, the South African, the Brazilian, and the Indian variants. Now they're called alpha, beta, gamma, delta. It also prevents any potential nationalistic antagonism that might result from giving variants certain geographic names. And these are the four that's currently, this is a screenshot from today from the WHO page. These are the four variants of concern, which is the second highest level of concern. There's one above this, which I think is a variant of high consequence, which we're not at yet. But variants of concern are basically variants that have shown that they can spread very broadly and can evade certain, either the immune system, transmit better, undermine certain treatments, etc. They're also what are known as variants of interest, which is kind of the minor league. So these are waiting in the wings, and they're currently being monitored, even though they're not spreading as much. The California variant used to be a variant of interest. It was called Epsilon, but since been demoted. It is no longer a variant of interest because it didn't spread, but the idea behind the California variant is encapsulated within the Delta variant. So this is the world of variants. It's basically a bunch of mutations in very important areas of the virus that have very severe implications for how effective the virus is. Just finishing this section of the talk, if anyone's interested in actually entering this rabbit hole of variants and viruses and mutations, there's a great website called nextstream.org. I have nothing to do with them, but it's actually a very good website. And you can start seeing actual sequences that were found. And you see that there's new mutations all the time. Most of them do not help the virus, but some of them, as we see, do. Okay, so that's all about viruses, mutations, and variants. Let's now go to the Delta variant. So as we said, this one emerged in India in late 2020. Its official number used to be B1617. There's actually three versions of it, 1, 2, and 3. The one that we're interested in is number 2, that's the Delta one. Number 1 is a variant of interest, kind of a minor league variant known as Kappa. And number 3 has not received a Greek letter yet. This is as of today, certain countries where the Delta has been officially confirmed through genetic sequencing. You see Israel's one of them. It doesn't have a lot of cases, but it is experiencing a wave, as we'll see. This is a geographical view of where the Delta variant has been most prominent. This is taken from Wikipedia. Okay, why are we so worried about Delta? Well, number one, we're worried about it because it seems to be the most successful variant currently. And successful is in quotes, successful for the virus, very unsuccessful for us. The virus known as Delta, the Delta variant of the coronavirus, is spreading very rapidly around the world. It has already been noticed in over 100 countries. It's the dominant strain in places like India, America, Israel, right? So a vast majority of new infections are Delta, the UK as well, and many other countries as well. And the reason that it's become the dominant strain is that it's so effective at infecting. It's very transmissible. Some people put estimates, and we don't know yet, and you're going to hear that a lot from me in the next few minutes. It's a new strain, and it's newly prevalent around the world. And so a lot of the questions that we'd like to have answers to, we have preliminary answers to. We don't have final answers to. And scientists are working very hard to get those answers. But I want to make sure that people understand that a lot of these things are still very preliminary. But here's what we think we know. We think we know that this is very transmissible, perhaps even 1 to 2 times more than the original strain, which in the exponential world of viral transmission can be a very powerful multiplier. We think that it's more resistant to certain antibodies. In other words, if someone was infected with a previous strain, they may or may not have as strong of protection to this new strain. It may cause higher severity. One report found that hospitalization risk was almost double that of the alpha variant. There's still a lot of questions around those results. And we're even seeing a different clusters of symptoms. So we're seeing people report cold-like symptoms relating to headaches, runny noses, and sore throats, instead of what we got used to in the first year of COVID, which was loss of smell, shortness of breath, fever, and persistent cough. So we're seeing a lot of those. Next, some more results hot off the press, and these are from preprints that have not yet been peer reviewed, and I'll emphasize that a few more times as we get to these studies, but this is kind of on the list of what we think we know about Delta. There's some preliminary unpeer reviewed results from China that seem to indicate that there's a much higher viral load in people newly infected with the Delta variant. And what that means is that if you literally count the number of virus particles floating around in the cells or blood of an infected person, you see many, many more a few days after infection than you would otherwise. And that means that the virus is much better at doing the whole exponential multiplication thing within the host cells. And so that is still very preliminary, but 1,000 times the number of virus copies compared to the original strain is obviously a very big difference if it turns out to be true. And as a result, we're seeing people are being reported as becoming infectious sooner. So in other words, it takes them 4 days to reach detectable levels inside a person, whereas previous strains, it might have taken them 6 days. And this is because the virus is so successful at copying itself within the host. Okay, so I'm just going to briefly show you. Here's that study that I mentioned. This is MedArchive, which is a preprint server. This means that pretty much as long as the paper is well formed and meets certain standards, you can submit it to MedArchive and it goes up on the web, even though no scientist has blessed it as being correct or thorough. So this is very much that. This is a preprint, and it's out of China. It's the one that found that it was about 1,000 times higher viral load and faster replication. Here's another one that looked at household transmission, and it found, here's the magic number, household transmission was 1.64 among Delta variant cases compared to Alpha cases. So in other words, it's transmitting much faster amongst members of certain households compared to the Alpha variant, which is again the British variant. And if you recall, the British variant, when it showed up, everyone was emphasizing how fast it was transmitting compared to the original variant. So if the British variant was faster than the original strain, here the Delta variant is faster than the British strain. So we're now kind of entering the next gear of transmission speed and efficiency, which is obviously not a good thing. Okay, so one of the big questions that people are asking is, what is the vaccine effectiveness against the Delta strain? We're going to get to Israeli answers to this as well. But a number of recent studies, which we unfortunately don't have time to go through here, but they've been published in very prominent journals, New England Journal, Lancet, and also some preprints published at MedArchive, have found that fully vaccinated individuals maintain strong protection against the Delta variant, especially to prevent severe illness and hospitalization. But some studies have found that partial vaccination, meaning people have only gotten 1 dose of a 2-dose regimen, or previous natural infection, individuals who had gotten sick with, let's say, the original strain or the British strain, now known as the Alpha, their antibodies, their immune system was not sufficiently protective against being infected and getting sick with the Delta strain. So that's good news for vaccinated individuals who are fully vaccinated, because there's still strong protection, at least against severe illness, according to the studies that have come out so far. But it is also a clarion call to encourage full vaccination, even amongst, well, especially amongst people who are unvaccinated or have only gotten 1 of the 2 doses, but also to encourage vaccination amongst people who are previously infected and have not had any vaccine. So I want to emphasize that this is not medical advice. Everyone should seek advice only from their licensed medical professional and their doctor. So this is a scientific talk where we're reporting non-final recommendations that were identified in recent research papers. So please do not take anything that I say here as medical advice. Okay, so with that said, we'll keep moving. This is a paper that was out today. It was published on MedArchive a month ago, and it came out in the New England Journal today. And it's representative of many of the other studies. They basically found that if you had the Pfizer vaccine and you had 2 doses of it, you could prevent symptomatic infection with 93%. These are the numbers we've gotten used to seeing previously. So that was against the Alpha variant. And with the Delta variant, that number dropped to 88%, which is still sky high. So yes, there seems to be a slight drop in vaccine effectiveness, but this is not a scary drop, right? The vaccine that's 88% effective is still excellent amongst the Delta variant. This is the scientific name for the AstraZeneca vaccine, which is not used in America, but is used in England. The effectiveness of 2 doses was 74.5 for the Alpha, and it went down again a few points to 67% for the Delta. So we're seeing a slight drop in effectiveness amongst vaccinated individuals. And again, this is for the outcome of symptomatic infection. They reported much higher effectiveness for, and other studies as well have reported much higher effectiveness for preventing hospitalization and severe illness. What they did found was if you only had 1 dose of the vaccine, the numbers were not in your favor. So very important for people to get both doses. Okay, I'll quickly go through another couple of studies and we'll get to the Israel story. So this was an article that underwent accelerated article preview at Nature because of its timeliness. So the Delta variant was resistant to neutralization by some monoclonal antibody therapies. So I don't know if you've heard about this, but for example, when President Trump got COVID, he was given a certain therapy that was based on antibodies. So this study found that some of those antibody therapies were less effective against the Delta variant. Some were still effective. Sero, which means blood, from convalescent patients, people who had survived COVID and recovered, were 4-fold less potent against variant Delta relative to the variant Alpha. So that means that people who had gotten sick in the past year and had antibodies that resulted from that infection, their antibodies were less effective against the Delta variant. People who had received 1 dose as opposed to 2 doses also barely were able to get protection from that 1 dose against the Delta variant, but administration of 2 doses was sufficient and gave 95% response there as far as providing the antibodies that interacted with this Delta variant. However, they're noticing a weakening in the shield. So titers were 3 to 5-fold lower against Delta than Alpha. So it's still providing protection, but there's a dent in the armor, a chink in the armor. In other words, we're not moving in the right direction with this variant, and that is raising the scepter of potentially having to update the vaccines, which luckily is easier to do using the mRNA platforms of these new vaccines. So let's get to one more. So this is another New England Journal of Medicine study. And this one, again, is looking at Kappa, which is Delta's cousin. It's the other Indian strain. And they found, again, that people who had recovered from COVID-19, their antibodies were less good at binding with the Delta variant. Okay, and we mentioned previous things. Okay, so good, we're right on time. What we're gonna do in the next 15 minutes is talk about the Israeli story, and then we'll save the 15 minutes that remain for questions. I'm sure there's gonna be a lot of them. Okay, trying to squeeze a lot here in 45 minutes. Let's see if we can do it. So this is the planet on which we are. Here's the Middle East and Europe and Africa, and right there in the corner is Israel. First of all, what people don't know is Israel didn't only have a very successful vaccination response, but it also had a very severe lockdown regime during the first year. Here is the vaccination story, and then we'll see the lockdown story. So this is from April, a screenshot of a website that everyone seems to have added to their favorites recently, Our World in Data. And what we see here is Israel kind of jumping out front as far as the speed and penetration of its vaccination campaign. And you can see other countries catching up. Today, if you go to that same website, you'll see that a lot of countries have joined Israel with high vaccination rates. If you look at the light green bars, that's anybody who's gotten at least one shot, in other words, partially vaccinated. And if you look at the dark green bars, that's people who are fully vaccinated. So Israel, I think, according to this chart, is still ahead. There's a lot of countries that are not included in this list. And I think you can select on that website which countries are included, but I think this was the default list. So you see that Israel, amongst these countries, is leading in the fully vaccinated individuals, but there are other countries, Canada, United Kingdom, United States is also slowly getting there as well, with high penetration of vaccination. Here's what it looks like from inside Israel. The dark green here is the first shot, the light green here is the second shot, and this is the millions of people, right? So right now, Israel is almost at the 6 million mark out of 9 million that have gotten at least one shot. So that's a pretty good penetration. Here's a picture of when those vaccines were administered, and the little details here tell a fascinating story. So what we see here is Israel started its vaccination campaign just at the end of 2020. And the dark green here are people getting their first shot. These were the elderly, the immunocompromised, people who were very susceptible. And initially there was an emergency to get them vaccinated. And you see there's their peak coming in again for their second shot 3 weeks later. And then it attained a kind of slower clip of about 70,000 shots a day. And then kind of most of the people who wanted to get vaccinated were, and the numbers went down. And then it kind of stopped. Over here, there was the announcement that kids can now get vaccinated, kids over age 12. And very few parents took their kids to get vaccinated. There was hesitancy around that decision. And so another interesting story is this little green bump right here. This is basically a wave of vaccination of young kids, of 12-year-olds to 18-year-olds. And the reason that it happened was that Israel has had a batch of vaccines that was about to expire. In other words, they had tons of vaccines that they could, or not tons, but enough vaccines for everyone to get, but there was going to be an expiration date. So the government made a big campaign, says, if you have to get your first shot by this date, otherwise you won't be able to get your first shot and second shot because it'll expire. So that caused a lot of people to go and get it. And then the day after that deadline, the government announced, oh, and by the way, we just made another deal with Pfizer, so there's going to be no interruption to the vaccine schedule. So you should still come. So you see a very nice response to the deadline. And then as soon as the deadline went away, the number of kids being vaccinated resumed a very slow pace again. So interesting public health communication lesson learned there. This is where it stands today. We have over 90% of the population over the age of 60 vaccinated. And you can see for each age group going down to the kids right now, this doesn't include the 10 and 11-year-olds, so the numbers are naturally lower, but we're about at 40% for the kids. Okay, and this is Israel's new confirmed infections per day. So Israel's already had 3 waves, the first wave in March of 2020, and there's the second wave over the summer, the third wave, which happened this winter, and that was cut short by the vaccination campaign. And now we're seeing the fourth wave, which is credited to the Delta variant. And you're seeing right now, the numbers are around 1,400 a day of new cases. Here's the severely ill. So again, 4 waves, 1, 2, 3, 4. We're starting to see the beginning of a fourth wave. But to do a very simple calculation, which is extremely misleading, and we'll talk about why it's misleading, but if you look at this second wave right here, at a certain point, we're getting 9,000 daily confirmed infections. And we're getting about we had about 900 seriously ill patients. So that was about a 1 in 10 ratio there. Now we have 1,400 to 720, so 72. So it's about a 1 in 20. So it's about half, the ratio is about half as much. Now this is extremely unscientific for reasons that we're going to elaborate on in the next few slides. But the general message that's coming from the public health officials is, yes, there are a lot of new infections, but a lot of them are amongst kids, or a lot of them are very mild. Amongst vaccinated individuals, they're very mild. And so we're not seeing the extreme rise in severe illness that we saw last time. And again, that was a very unscientific analysis, but it drives the public conversation. These are kind of the analyses that people are doing in the media. Okay, and this is the number of new seriously ill patients per day. Again, you're seeing it's not at the levels it was before, but time will tell. And new deaths also thankfully very low. This is the R. Okay, here's another story. Israel started ramping down its testing rate and closing testing centers because people weren't coming. And now, as the Delta-induced fourth wave arrived, there has been a sudden resurgence of testing activity. And we see that Israel is now ramping it up because there have been some very long lines. Okay, so now in the last 8 minutes, we're going to get to some interesting issues that have arisen, and then we'll get to your questions. Okay, so the Israeli Minister of Health this week said the following sentence. He says, "We understand the coronavirus won't soon pass from the world. After the Delta variant, is likely that another variant will emerge in the world, and therefore our strategy is clear. Life alongside the coronavirus." So there's two very nice truths in this statement. The first truth is that there are probably going to be some more variants, and unfortunately that seems to be the case here. The Delta is probably not the last variant. I wish it were. There's probably going to be some more mutations, and coronavirus is not going to disappear anytime soon. And so since Israelis have been world champions of lockdowns this past year, there's little excitement in the Israeli population amongst the Israeli population about the idea of going back into lockdown. And so the messaging now is very focused on how can we live alongside the virus. So the current approach, so let's look at some of these things that Israel is doing. So one, they're trying to continue to promote vaccination of the remaining unvaccinated populations. This is difficult because there's a self-selection here. The people who wanted to get vaccinated or could be convinced to get vaccinated, they've been vaccinated a while back. And the people who remain unvaccinated are those almost by selection that have a reason not to be vaccinated. They are hesitant. They're fearful of it. They have other reasons for not wanting to do it. And so we're kind of plateauing, and there's diminishing returns. It's becoming marginally harder to convince the remaining people to get vaccinated. Israel's ramping up testing, and it's also introducing rapid tests. So rapid tests have been in use in other countries, and you can go to a drugstore and get them. Israel hasn't introduced them because it was worried that they weren't reliable enough. And now it realizes, okay, well, they're better than nothing. And they want to live alongside it. So they want to help empower the public to do tests. They're reinstating the Green Pass system. Today they announced, this is a recent update, that for all indoor events over 100 people, they will enforce that only people who have a Green Pass showing that they are fully vaccinated or can show a negative test, either rapid in the last 24 hours or PCR in the last 72 hours, or they have a certificate that says that they have survived COVID. In other words, they got sick and recovered. They're allowed in. But if you don't have one of those conditions, you can't go into an indoor event with more than 100 people. And they said if the Delta variant continues to increase the daily case numbers, they'll reduce that number to 50, et cetera, et cetera. Israel's reinstated travel restrictions to certain parts of the world. There is a whole scan. Israel pretty much has one gateway, which is Ben Gurion Airport near Tel Aviv. That's pretty much the only way in and out of the country. So it's virtually an island. And so the travel restrictions are a very big one because new variants obviously can either originate in the population, but since Israel is a small country, the chances that a new dangerous variant would arise here are low, just like anywhere else in the world. But they're more concerned about the rest of the world. Billions of people, it's more likely that something bad might happen just because there's larger numbers there. And so there's been a lot of focus on reinstating those travel restrictions. Ironically, Israel has shortened its quarantine from 14 to 7 days. And this is very confusing. Why is Israel shortening its quarantine period if cases are going up? And the answer is that they had seen that they had not found a good solution to enforcing compliance with previous quarantine restrictions. People said 14 days is too much. That's unrealistic. I'm not going to stay. I'm going to violate quarantine. I'm going to leave my house. The police say, well, we don't have enough personnel to track every citizen in the country. And so the idea was, if you reduce the quarantine to 7 days, maybe you'll get more compliance. And that's another interesting experiment going on now, and we'll see if it works. Israel's already started giving third shots to a very, very specific group of people. This relates to the booster conversation that a lot of people are having now, will we need a booster shot? Pfizer, as many people already know, is now applying for an update. It's applying for approval for an updated version of the vaccine that will be tailored to the new variants, including Delta. And in the meantime, Israel has given a third shot of the original Pfizer vaccine to people who are immunocompromised to boost their immunity. That gets into two questions. Number one, would a third shot make someone, especially immunocompromised, have more antibodies and therefore have a better chance of fighting Delta? Number two, is there something called waning immunity? Israel, since it was one of the first countries to have a rapid national vaccination campaign, many of the people who were vaccinated, especially the most vulnerable, were vaccinated in December 2020 or January 2021. Now we're in July, so it's been over 6 months. And we don't know this, there's studies ongoing to figure this out, but if there's something called waning immunity, which means that the vaccine works really well in the first few months and then less well after, which we don't have evidence that it does, less well after 6 months, then that third shot can maybe boost them back up. So there's an open question right now. Does immunity against coronavirus that is granted by these mRNA vaccines, does it wane over time? We'll get answers to that question in the next few months, but that is part of the booster conversation. And Israel's waiting and seeing about the booster shots. The talk is that there'll probably be a booster shot available, hopefully for the fall or winter. So that's the state of play in Israel. In the last 2 minutes, I want to talk about some studies that have come out. Now, these aren't really studies. These are in the food chain of scientific truth. There is peer-reviewed studies, which are usually pretty good. There's always some limitations that apply. Next, below that, the tier below peer-reviewed studies are these preprints, which are basically pre-publication copies of the paper that haven't been peer-reviewed. And so they're considered very questionable, not necessarily because the science is bad, but they just haven't gone through the verification process. And then below that is media reports that are just released to the media or leaked to the media. And it's very hard to know what scientific method was used. So that's kind of the bottom of the, if you will, scientific food chain. So recently, two such media reports came out. One of them was that health ministry data in Israel released in early July said the vaccine's effectiveness, this is the Pfizer one that was predominantly used in Israel, which was over 90% before Delta, is now at 64%, which is a drastic drop, right? And what we saw in that New England Journal of Medicine paper, which is based on British data, it had gone from 93 to 88, if you remember. And here it's 90 to 64, a much more precipitous drop. So that raised a lot of eyebrows. And alongside that New England Journal of Medicine paper, there were 3 or 4 other very kind of established groups that had gone through peer review that found similar data. So the outlier is here. This is not what most or almost all of the other studies found. So this raised a lot of questions, which is, why is Israel getting different results than the rest of the world? 2, and this just happened in the last couple of days, a Ministry of Health, there was a media report about a Ministry of Health meeting where data was presented showing that the immunization's effectiveness in preventing serious illness is at 80%, down from 95 or so. And for the elderly, it's down to 50%. And this raised so many eyebrows that all the media report, they interviewed all the professors and all the scientists, and they tore it to pieces. They said, this is a media leak, and it's completely unscientific. And what's interesting is the criticisms that were leveled, and we'll end with this slide, are very revealing of some interesting challenges going on. So I saved the worst science for last, just to show that it is important to rely on the scientific process and not to look at media reports. But let's look at these criticisms. So the first thing that people said are, okay, these are media reports based on leaks. And even if they did do a good analysis, they did it based on very small numbers of people. Okay. Number 2, there's confounding between vaccination and testing behavior. It's a really interesting point. If someone got vaccinated, that person probably takes coronavirus pretty seriously. And so now, if they're worried that they might have been exposed, they'll go and get tested because, again, they take coronavirus seriously. If someone does not take coronavirus seriously, they probably didn't get, or they may have not gotten vaccinated earlier. They're still not vaccinated, and they're probably not going to go out and get tested either. So there's likely a lot of under-ascertainment, in other words, lack of testing of the unvaccinated group, which means that we're missing a lot of the infections in the unvaccinated group, which means that it makes it look like the vaccine isn't so effective because look, compared to the unvaccinated group, the number of infections here is not that much lower. And the reason we're seeing that is because we're missing a lot of the infections in the vaccinated group. The second one is also an interesting point. Because of Israel is so highly vaccinated in the elderly and in the adult population, the definition of serious illness, which was used until now, was basically a drop of oxygen saturation in the blood below a certain level, which shows that that there's a severe illness going on. So it turns out that before almost the entire adult population was vaccinated, this was a pretty good marker, because if your oxygen saturation dropped below a certain point, it stayed low, and you had a rough patch there, a rough bout of illness, which unfortunately in numerous cases ended in death. Amongst vaccinated individuals, if your oxygen saturation drops, it does so for a very brief level, and then your body, using its vaccinated protection, bounces back very quickly. And so these individuals, even though they had a very, very short bout as their body was fighting the virus, but then they kind of got back up, are considered seriously ill, even though they're not actually seriously ill. So due to the dynamics of the vaccination campaign, we have to update our definitions of serious illness so that we can actually get accurate representations of these things. So I quoted these kind of media reports at the end of the talk to show that you have to be very careful what you listen to these days. So with that, we're going to wrap up this part of the talk and open it up to questions. Ken, do you want to take it over?
Great. Ben, thank you for taking us through so much information in such a short period of time and so clearly. Let me get your sense of which direction things are going there. So there's a paradoxical phenomenon, perhaps, that the higher the prevalence of the virus is in a population, the less efficacious the vaccine is going to be, just when you need it the most. So these numbers that we saw, where there was a dent made, unfortunately, in the wrong direction in vaccine efficacy by the Delta variant, and we see rising cases and rising hospitalizations, are we going to see, in your gut sense, a diminishing efficacy of the Pfizer vaccine against Delta in Israel initially under those circumstances?
So it's a very good question. And I think the big challenge is going to be, even if we do see one, what's a good way to measure it? Because in a lot of these age groups, we don't have a lot of unvaccinated individuals. So, in order to do a classic kind of real world data controlled study, you need two groups to compare, one vaccinated, one unvaccinated. You want to control for all sorts of factors, and then you want to kind of put them in the wild, as it were, in society, and see what the infection rates are in both. It's actually really hard to do that study now in Israel because so many people are vaccinated in the older groups, and the people who are unvaccinated are so self-selected by this point. In some of these age groups, you're looking at over 95% penetration, and so the people who are unvaccinated are either they medically can't get it, or they're so strong, or they have such a different lifestyle or attitude towards this that they become uncomparable. There's no exchangeability in the words of the epidemiologists between the two groups. So that's one of the challenges. The second challenge is that if you were to find a drop in vaccine effectiveness, you wouldn't know why it is, because there's so many possible reasons. Here's another spanner in the works. The people who are most vulnerable, justifiably, were the first people to be prioritized for the vaccine: elderly, diabetics, etc. So in late December, early January, those were the folks who were getting vaccinated, hundreds of thousands of people a day. They are the ones who've had the vaccine the longest. They are the ones for whom the most time has passed since they were vaccinated. And so as a result, it's, there may be the waning immunity issue. And so all these factors make studying the changes in vaccine effectiveness due to the Delta variant very difficult. Is it for the reason that you mentioned? Is it for any of these other reasons, or even further reasons that we didn't have time to mention? So those are some, I know of a bunch of ongoing studies now, and the challenges to those studies, the questions, the scientific and methodological questions being raised are actually fascinating, because in a certain way, the Delta variant, is catching the world off guard. In another way, certain countries like Israel, because of their very successful vaccination efforts, actually make current epidemiological studies more difficult to run. So there's a lot more question marks than answers at this point, but they're interesting question marks.
Well, thank you. That was a nuanced answer. And it gets at this issue that the efficacy rate that's so widely reported, there's a lot packed into that number, and it means certainly not what most people think it means. If you're a 94% efficacy of the Pfizer vaccine, it does not mean that you only have a 6% chance of getting COVID no matter what you do, which is, I think, probably the way most people would interpret that number out there. And you helped tease apart some of that nuance. Obviously, and things are changing, and it's becoming even more complicated, and that particular metric is likely to be replaced with others. So now let me go to the chat and the Q&A and pick some questions. And here's one that's, I think it's a very straightforward question, but I think we can answer it because it's of general interest. Is there any evidence that COVID tests would react differently to the Delta variant or specific tests? In other words, are we not, because I've seen this come up in a couple of places in the questions, do our tests work for Delta variant?
Okay, great question. So the first part of the answer is we have no reason to suspect that the PCR test, which is has become the gold standard until now, to date, is any less good at identifying the Delta variant versus others. The reason being that the PCR test can look at all 30,000 base pairs of the viral RNA, and the Delta variant, has 20 mutations out of that. So there's still a lot of virus that hasn't changed that you can still grab onto. So from that perspective, there's no real concern. The greater concern is perhaps the timeline. So a study that came out a few months ago, an interesting study, which had some issues, but still revealed some very cool points, showed that the PCR test, which everyone looks at as this great arbiter of truth, if someone is infected on Sunday, on a given Sunday, and you give them a PCR on Monday, the chances that that PCR test will pick up the fact that they've been infected are 10 or 20%. I forget the exact number. And that number slowly rises until day 4 or 5, and only in day 5 do you get up to 90%. And so there's a whole incubation period in the body where the viral load, as it's called, the number of viral particles, is still very low. They haven't they haven't taken over the body enough to be noticeable on a PCR test, where you sample perhaps the nasal passages. And so the question is, perhaps the Delta variant, because it seems to replicate more efficiently and even faster in the body, perhaps the PCR tests are even better at capturing the Delta variant, because the Delta variant is very good at generating lots of copies of itself. And so the effect might actually be the reverse, that when we see higher infection rates now, maybe there were also high infection rates back then. This is a partial explanation, it's by no means a full explanation. But it could be that the reason we're seeing, some part of the reason that we're seeing such higher rates now is that the PCR tests are better at picking up the Delta than the other one. So again, that's all speculation at this point, but it could go either way. But it's a great question.
Great question, great answer. It's very complicated. The levels of virus in secretions are so high that several of the folks who develop these tests that I've spoken to, they're even concerned about false positives. Someone goes from one patient to another in an ICU doing testing. A tiny, tiny, tiny amount of contamination in the test collection process can produce false positives. And so, yeah, so we're really looking at some very interesting issues. And does having virus in your nose mean you have COVID in the first place? You're much more likely to have virus all over the place if you've been near someone with that many secretions. Here's another good question from the chat. There are many, we won't get to all of them. Apologies for that. How many people hospitalized from Delta variant are unvaccinated individuals compared to vaccinated? Specifically around some of the data you showed about the 1 in 20
I'm so happy that that question came up because I wanted to get to that point during the talk, and because of time we didn't get to it. So thank you to whoever asked that question, and thanks Ken for picking it. One of the big claims that I hear a few times a day is the vaccines don't, quote, the vaccines don't work against a Delta because look how many vaccinated people are in the hospital and even dying from coronavirus who were fully vaccinated, 2 doses, approved vaccine, and they got Delta and they were either seriously ill or, God forbid, died. So end quote. So that is the thing that I hear a few times a day. So here we have to kind of understand the statistical background to that. So let's look at a place like Israel. Israel, for elderly people, let's say over 60, we're around 90–95% vaccinated. Okay. So imagine you have 100 people, and out of the 100 people, 95 are vaccinated, 5 are not. And that's in the population, okay? And then you go to the hospital wing, and you see that in the hospital wing, there are 4 people who are severely ill with COVID. 2 of them are vaccinated and 2 are not. What does that tell you about how well the vaccine works? Well, some people might say, oh, look, half the people in the hospital wing with severe illness are vaccinated and half are not. Therefore, it's a wash. The vaccination doesn't even help. So that's obviously wrong. The right way to look at it is, oh, wow, out of the 95 people who were vaccinated, only 2 ended up in the hospital with severe illness. And out of the 5 people who were unvaccinated, 2 out of them ended up in the hospital. In other words, 2 out of 5 is 40%. In this cartoon example I'm giving, 2 out of 5 is 40%, and 2 out of 95 is 3%. So unfortunately, people don't run that statistical calculation in their head, especially when a lot of the people who are severely ill now are in fact the most vulnerable and elderly. And in America and in Israel, the sectors of the population that are most highly vaccinated are indeed the elderly and the most vulnerable. And so the argument that I just put forward, which I hope was understandable to people, that you got to put the right denominator around these things. And the other thing to realize is even for the original strain and even for the Alpha variant, which we used to call the British variant, no one ever said the vaccine is 100% sure to prevent someone from going to the hospital or prevent them dying. The numbers were always around 95%, et cetera, so somewhere around there. And those numbers from the controlled studies that we've seen so far, published in the New England Journal Today, in the Lancet, and the other kind of thorough studies, not the media reports that are leaks, show that the protection of the approved vaccines against severe illness or death or hospitalization seems to still be in the high 90s with the Delta variant. So the message that comes out from that is, yes, the Delta variant is a major public health challenge, and yes, the currently available vaccines are extremely effective, still extremely effective in protecting against the worst outcomes for those. So the one message, if anybody takes any message from this, is that yes, the science clearly shows that the vaccines are still very effective at saving lives against the Delta variant. So if you know someone who's hesitant and now says they don't want to get the vaccine because, quote unquote, it doesn't work against Delta, that's demonstrably false, and their life depends on doing the statistics right in that case.
One last question that I'm going to ask you to answer very briefly, and if you need to forward point to data that's coming on this, that's great. But this is another $1 million or 4 million shekel question, as it were, for you. Is there evidence of transmission from a vaccinated person to another vaccinated person?
Excellent. The short answer is no. And the longer answer is it's hard to tell because who transmitted it to whom, especially with something so transmissible, and people are not in bubbles, right? But I can say that there are a number of studies going on that are looking at household transmission, which can start giving us some evidence to help answer that. But it's very hard to tell who transmits a virus to whom unless the person is in an absolute hermetic bubble where the only person they come in contact with is another individual. So very hard to answer that otherwise, but stay tuned.
Great. Ben, fantastic talk. This questions that we got today really are different than what one has from a usual scientific talk. These are questions from a community. We're all together. We all are wondering how this impacts our lives. We're wondering what we should do. You've given us a lot of information in that direction, and we hope to continue the conversation with you, Ben, and we'll end it right on time. Thank you very, very much.
Yes, and thank you. And to anybody whose question we didn't get to, feel free to email me at that email address, ben_rice@harvard.edu, and I'll try to answer you through there. But thanks again, Ken, for setting this up. And thanks again, everyone who attended.